Good afternoon, New York, and the rest of our listeners around the globe. My name is June Stoyer, and I'm the host of the Organic View radio show. Our podcast is available on iTunes, Zune, and all major podcast providers. So if you can't catch the show live, you can download it or simply use our free podcast player, which is available on our website at www.theorganicview.com. If you'd like to connect with us, please post a question on our wall on Facebook or send me a tweet at June Stoyer on Twitter. If you'd like to be on the show or would like to find out about sponsorship opportunities, please contact us at questions@theorganicview.com. Today's show is sponsored by Austria's finest naturally authentic pumpkin seeds and pumpkin seed oil from the Stoyer mark, available at organicuniverse.com. Listeners of The Organic View can receive $1 off their purchase by using the coupon code ORGVIEW. That's ORGVIEW.
For other promotional offers, please visit theorganicview.com's website.
On today's show, Dr. Chris Connolly from the University of Dundee is going to talk about his fascinating new research about bee brains and colony health, which is jeopardized by pesticide exposure. So I'd like to welcome to the show Dr. Chris Connolly. Good afternoon, sir, and welcome to the show.
Thank you very much for the invite.
Dr. Connolly, could you please share with our audience a little bit about yourself?
I'm a neurobiologist working in University of Dundee, mostly interested in human disease, problems of the brain, how information travels around the brain to allow us to interact with our environment. So not really working on bees at all historically. So my interest in the bees really became because of the debate, the media, the work you've done and other people like you have done to bring the story to everybody's minds. And it brought it to my mind, and it made me realize that what I was working on in terms of mammals, of humans, was very similar to what we could see with the bees. We use the same transmitters. We still have brains. We have to interact with our environment. We have to learn, remember, communicate. So actually, it wasn't a big jump for me to move to a bee brain. And when I realized that one of the threats were these neonicotinoids, I thought this neonicotinoids is basically insect nicotine. It does for insects what nicotine would do for humans. And at high levels, it would just kill you. Lower levels, it might actually affect your brain function. So this is really where I came in to see, does the neonicotinoid actually do what it says on the tin to bees? So does it disturb their brain function?
Can you give us a little bit of background about the experiment and also which pesticides were used?
The big debate is on how much exposure of neonicotinoids occurs to bees. Everybody's been arguing over this for so long, so we decided to go directly to this, and we looked at two neonicotinoids, imidacloprid and clothianidine. So what we needed to do was to show that the neonicotinoid could actually reach the brain at a level where it could be neuroactive and disturb brain function. So to do this, we worked on bumblebees in this case. We fed the bumblebees sugar syrup at the level of neonicotinoid, imidacloprid, that's found in the environment. So that's 2.5 parts per billion we were fed. Now, over 42 minutes, we could just about detect the neonicotinoid reaching the bee brain, but it was too low a level to have an effect. So this is good news, because it means that the bee, when it consumes the nectar, would still be able to find its way back to its nest. It wouldn't work that fast. However, over 3 days, the level now accumulated up to 10 nanomolars, which is actually 2.5 parts per billion. This is quite a high level in the bumblebee brain. What does it do? And so we investigated looking at the bumblebee brain cells to see if their function was disturbed. And in the case of clothianidine, at this level, the brain cells could no longer produce energy. So basically, they just stopped. And this is the dose after 3 days of clothianidine when applied to the brain cells immediately. So it's an immediate block. Now, interestingly, imidacloprid, when we did the same thing at the same concentration that's present after 3 days, it didn't do this. But then we looked to see, does it make the bee brain cells more vulnerable to other insults? So the brain cells normally communicate, they talk to each other by releasing another neurotransmitter called acetylcholine. So we looked at acetylcholine itself, and we found acetylcholine does not damage the brain cells, as you'd expect. However, after 3 days of exposure to imidacloprid, these brain cells were now vulnerable to normal transmitter, and then they stopped producing any energy and the cells stopped working. The really amazing thing was at 1/10 of that dose, this is 1/10 of the field realistic dose, the same thing happens. And this is to imidacloprid. We didn't go back and look at clothianidine at these super levels because it worked quite well directly without any need to make cells vulnerable. But one could anticipate, because clothianidine's 10 times more potent, it might be have the same effect long term. So maybe clothianidine will work at 100 times of environmental dose. So this is quite striking. So this shows that in the brain of the bee, the pesticide gets there at a level that does perturb its function. And this is after being fed at environmentally realistic doses in just 3 days. Oilseed rape will flower for 3 or 4 weeks. Plus, the neonicotinoids can build up in the soil. People have found it now coming up into wildflowers, dandelions, etc. So the length of time at which exposure could be occurring in the wild may be much, much longer than 4 weeks. We don't know that. That's a place where we need more evidence. But 3 days is enough to affect a bumblebee. Now, of course, that's one single bumblebee. Bumblebees, of course, live in nests of up to about 200 bees. And the real output of a nest is the queen, her ability to lay eggs and produce bees and produce next year's queens. So really what we need to look at is how does a colony be affected by this kind of exposure? So we then took some bumblebee colonies, we took them to a remote part of Scotland, the west coast in Wester Ross, which is near the Isle of Skye. There's very little pesticides used there. So this provides a good environment where you can say what is the impact of this pesticide when fed to the colonies at the field realistic dose of just 2.5 parts per billion. And so we did this, and what we found was quite shocking that the bumblebee colonies that were fed the imidacloprid had about half as many bees, half as much brood. The nest was in terrible condition. It was overrun by fungus, invaded by wasps. So there was a clear distinction between bees that were not treated with pesticide and those that were in a complex environment. And the complex environment is an important point, which I could maybe come back to later. So combining bees feeding on sugar syrup containing field realistic levels, it gets to the brain at the dose that's high enough to affect brain function. Other studies have already shown it affects the ability to learn, remember, and affects whole colonies. So we then confirmed this ourselves on whole colonies, and bingo, it was easy, quite shockingly easy to find the problem.
Moving forward, what is your next subject for research going to be?
Well, we now are interested in looking at how multiple threats can work together. So we already know in medicine that if you treat patients with multiple drugs, then actually this can have unforeseen problems on human health. In fact, I have a friend of mine who's a consultant at Dundee. He recently had a patient that they could no longer help by medication, and the patient was on 12 or 13 different drugs. They had to reluctantly withdraw the medication and let nature take its course, and nature did take its course, and the person recovered completely. So this is an illustration of what multiple chemicals can do that you can't expect, you can't foresee it. Now, in the UK, we have hundreds of pesticides that are licensed for use. The use on a local scale are not recorded, even though EU regulations say that they must be recorded. The governments don't collect this data. The potential risks for multiple exposures of different pesticides that may work together is enormous. So this is an area that we think we need to look at, that how do chemicals work together to create either bigger risks or new risks that we didn't even know existed. So I think this is an important area that's not tested by safety. Each individual pesticide is tested for acute and chronic toxicity. Obviously, it doesn't go far enough because it didn't pick up the bumblebee toxicity that we see in colonies. And the UK is looking into how we can maybe cover that knowledge gap in the future. But basically, it isn't possible to predict how numbers of chemicals can work together. And so this is a major flaw in safety. It's not anybody's fault because nobody knows what tests should be done to try and identify it So this is what we want to work on, to try and find molecular indicators that might tell you, flash up a warning signal to say these combinations might not be safe. After all, we have lots of idiopathic diseases in humans. So idiopathic means diseases we don't know the cause for. So it's possible that combinations of pesticides might actually cause certain idiopathic diseases. And although it's very brash to say that out, it's actually impossible to disprove it also. So this is an area that I think we urgently need to understand if we're going to continue to use hundreds of pesticides in our environment to try and combat what we consider is a problem of food production. So until we can actually move away from pesticides, we need to very quickly understand how we can learn to live with them.
I couldn't agree more, especially since neonicotinoids are mobile in groundwater. And if you think about the impact of one particular chemical on any given species and then combine all of the different possibilities, it's going to be a very interesting study, to say the least. I'm sure it's going to open the eyes of many people.
I hope so. I think apart from knowing which combinations might be really dangerous, it would at least also be quite helpful to know which combinations aren't or aren't predicted to be. So at least we can be sensible about what we do with pesticides until we can reach a time when we don't need them at all, which is obviously everybody's real goal. But in the meantime, we need to learn to be intelligent about our use of pesticide, to do as in medicine, look at a risk-benefit. How much benefit do we really get from a pesticide? How much risk is there really from that pesticide, and can it be justified in that situation? At least then we could probably dramatically reduce our pesticide use if we could just stick to those simple principles, if we had the data to support the knowledge base that we need to make those decisions, of course.
Dr. Connolly, that's a very interesting point. When it comes to pharmaceuticals, there are mechanisms which can assist a patient, especially if different drugs are prescribed, to alert the pharmacist or even the patient that the particular combination of the drugs prescribed or being administered, maybe it's two different doctors, may wind up creating a complication that was unforeseen. So the research that you're proposing really does make sense, especially from that perspective, since we don't know what these combinations could result in.
Yes, and the users wouldn't need to understand the science. If we could, say, attach a color or a number to a particular risk so that any chemicals that are labeled red mustn't be used together, then it would be a very simple system for people to understand. But really we need governments to provide the money to try and provide this kind of high throughput molecular approaches to try and get the information in the first place. But money's tight everywhere. It's very hard to get money to do this kind of research. But this is desperately needed.
When it comes to our health, as well as the health of the environment, I think that should be a priority, and hopefully the powers that be feel the same. Dr. Connolly, thank you so much for your time. Do you have any particular message that you'd like to leave with our listeners?
I would like them to consider what I mentioned about risk and benefit that we use in medicine. So if they consider using pesticides in their garden, please consider what the risk might be to your pets, your insects, the pollination, and also what the benefit is. I found if you leave things alone, they find their own balance. So think carefully. It's very hard to justify the use of pesticides in gardens, I think.
Dr. Connolly, it has been a pleasure having you back on the show, and I would love to invite you back when you are finished with this new research.
Thank you very much.
Oh, you're very welcome. And folks, please check out the companion article that will accompany this interview on theorganicview.com. Thank you for tuning in. This has been June Stoyer with the Organic View Radio Show. Have a great afternoon, everyone.